Hello, good afternoon, dear students. Sorry about the delay. All right. I can see the centers online. Sorry, I was a bit off. Okay. So, hello, Shillong, and hello, St. Paul's. Hello, Baptist School. How are you? Okay, so we are beginning now. Centers, please report your attendance. Please report your attendance. Okay, so in the last class, we have covered photosynthesis, we have covered respiration. Okay, so the major parts of class 11th, which have a good percentage of questions in in the NEET examination, like morphology, anatomy, the plant physiology, these portions are almost over. Now we need to do some other topics as well. Okay, so these topics must be covered because they are fairly easy, but they require some conceptual understanding. Okay, so because of that, we are going to study it. So today I am going to start with plant growth regulators. Plant growth regulator. Okay, plant growth regulator simply means plant growth hormones. Plant hormones. Okay, so plant hormones are the things we are going to discuss here. All right, so we are going to study the plant hormones here. Now this requires a bit of conceptual understanding about how the hormones work, okay, and where they are synthesized, what is their mode of action, what effect do they have. Okay, so these things must be taken care of. So plant growth regulator, you know the definition. These are plant hormones, and there are namely 5 hormones. Okay, auxin, cytokinin. Okay, and we have abscisic acid. Okay, and we have gibberellin. We have a gaseous hormone also called ethylene. Okay, so these are 5 hormones we need to study. Please note that these are not the only hormones, but these are the major hormones. There are some minor hormones as well. Okay, they play a special role in some local situations. For example, if a plant tissue sample is damaged, a plant leaf is damaged, these plants will release a chemical which tells the other cells the sensation of pain. It's a chemical sensation of pain. Plants do not feel pain in the same way as we animals do. Okay, that is the major difference. They do not have the nervous system, but they have the chemical coordination system. Okay, so there are some local hormones, but we do not need to know about them. We do not need to study about them. We are going to study the major hormones and their effects. Now this is a very important part, so please pay attention to it. Okay, so first of all, we are going to cover about auxin. Okay, so there is a nice story about auxin. I hope you will all like to listen to it. Okay, auxin was, you can say it was the first discovered plant hormone, and it has some specific effects. Okay, it was isolated from human urine. Okay, and effect on plant is it induces tallness of plants. Okay, but because it was isolated first of all from human urine, so please do not think that if you go out in the open and irrigate the plants. Okay, there are some people who irrigate the plants, okay, in ways we do not want to imagine. It does not mean that the plants which will receive the urine will also get auxin, so they will grow taller. Please do not think like that. Okay, it's a very minor concentration. Okay, it's not sufficient enough for the effect we want to observe, but yes, it produces a minor effect on the tallness of plants. But anyway, after this discovery, there were some other experiments on auxin. For example, Darwin and Charles Darwin and Wendt performed some experiments on phototropic effect of oxygen. Okay, phototropic effects of oxygen. Now, what does phototropic effect mean? Suppose this is a shoot axis and I am putting it in a cardboard box. Okay, I am putting it in a cardboard box, and in this cardboard box, suppose light is coming from this side only. I am making a cardboard box and I put a small hole here. Suppose this is only one side of the cardboard box and there are other sides of cardboard box, and on this side I put a hole and light is coming in into the darkness. So the plant will bend towards the light. All plants, when growing in a shady area, they try to bend towards the light. This is called phototropic effect. Now, what is the reason of phototropic effect? This was investigated by a scientist called Wendt, and what he did was he put some experiments. Now, let me tell you the reason first why oxygen causes this effect. Oxygen has an effect on cell elongation. Okay, oxygen has an effect on cell elongation. If cell is elongated, cell length will increase. So that is the effect oxygen produces. How it produces this effect? It is because oxygen causes loosening of the cell wall microfibrils, and because the cell wall has loosened, the inner part, it will rapidly absorb materials. Okay, and another thing is it causes some changes which like absorption of water and minerals from the outside. So the cell will absorb water and minerals. First it will absorb minerals, water will follow. So the overall cell volume is going to increase. Okay, so cell volume has increased, cell length has increased. This is the effect of oxygen. Okay, so if there are two cells, one receives oxygen, the other does not receive oxygen, the cell which receives oxygen will grow taller and more in volume. Okay, so what happens is oxygen has a property, it escapes from light rich side to light deficient side. Okay, suppose this is a shoot axis and it is bending towards the light. Why is it bending towards the light? Light is coming from this side. On this side, on this portion which faces the light, oxygen has escaped from this side to this side, okay. So more growth will be on this part. More growth will be on this part. If more growth will be on this part, this will bend towards the light. Why, you will say, then why does it bend towards the light like this? Because oxygen is more in the top region. Oxygen is produced by the apical meristematic cell region. So more oxygen here. So upper part will bend more than the lower part which receives less oxygen. So there is a curvature. Okay, this is the effect. Now, Wendt had performed some experiments. We must take a look at that, and these experiments are usually drawn in the diagram, in the exam in form of a diagram, and you need to know about it. Okay, so let's take a look at that. All right. So how does this produce an effect? A scientist called Wendt performed some experiments on oat seedling. Now I have told you that oxygen is produced in the top part. So this is I will shade the top part which produces oxygen. This part. Okay. So there is oxygen being produced in this part. Oxygen produced here. Now I am subjecting this oat seedling to unidirectional sunlight. unidirectional sunlight. Now what will happen? This oat coleoptile will bend towards the light part. This is called phototropism. Okay. Oxygen will escape to this region. Oxygen will escape to this region. More growth on this side. More growth on this side. Okay. Less growth on this side. Less growth on this side. Okay. This is called phototropism. Now there are many things. I suppose that this is something. Do you find this boring? Is all who is saying this? Why our chapter is so boring? Okay, I understand. Plants are boring, I suppose. Okay, all right, if you think so. But you have to study it. Okay, if you don't study it, you will be at a loss. I do not want. Look, I do not want to subject you people to torture. Okay. All right, not plants. Okay, so what are you saying? But our topics. You can suggest topics. Look, there's not much is left in this part. After this class 12 part, reproduction will start. Okay. Now my part is sexual reproduction in flowering plants. Ma'am will teach human reproduction and all that. And then you have the genetics. You can choose who teaches genetics to you. Okay, but probably I will be taking it, or maybe ma'am will take it. I'm not sure very well. But these are topics you have to study. Okay. Now let's. You were talking about something interesting. Okay. So oxygen has a very particular effect. Okay. You have seen a lotus flower opens at the sunrise. Lotus flower has the property of opening at sunrise and closing at the dusk part. Okay, at 5 or 6 p.m. it closes. Why does it happen so? And sunflower will always face the sunlight, the sun. Okay, so why does this happen? Let us study the reason. In a lotus flower, I told you, in any case, oxygen will always escape from light side to the dark side. Okay, it will always go towards the dark side. So suppose this is a lotus flower bud. In the morning, this is closed. Now, when it is closed and light starts coming in, oxygen will go towards the inner side, okay, towards this side. So more growth will be on the inner side. So the flower will open. Okay, so the flower will open. Now during the daytime, when this part of the petal is exposed to the sunlight, now oxygen will go towards the back side. When more growth is on the back side, the flower will close. Okay, so you can say that opening and closing of lotus flower is because of oxygen. Okay. Sunflower always faces the sun. Why does it happen? Because suppose sunlight is coming from this side and sunflower is in this position. Now on the back side, oxygen will always escape towards the back side. Now sun always changes its position, so the back side oxygen will always escape to the back side of the flower. If there is more growth on the back side of the flower, so the flower will always keep on facing towards the sunlight. Okay, so sunflower always faces the sun due to oxen. Okay. There are more effects also. I can tell you about it. All right, but you can write it in general way. Very short points you need to remember. These will be useful in the exam. Okay, so they are opening and closing of flower, opening and closing of flower is due to oxen, or any flower bud is opening due to oxen effect. Now there is one more interesting thing. You go in a garden, the gardener always cuts the top part of the plants. He always cuts the top part. He always trims the top apical bud of the plant. Why does he do it? Because oxen has the property of apical dominance. Oxen shows apical dominance is due to oxen. Apical dominance is due to oxen. Okay. Apical dominance is due to oxen. After that, apical dominance is due to oxen. If apical dominance is due to oxen, then what does it mean? If we cut the oxen is synthesized in the apical meristem, so if we cut the top part, then apical dominance will end and the lateral branches, plant will acquire bushy habit. Okay, it will acquire bushy habit. Okay, gardeners cut apical buds to reduce apical dominance, and if lateral branches will come, plants will appear more green. So whenever you go in a garden, there is a lot of greenery. Why? Because the gardeners are regularly taking care of the plants. They are cutting the apical buds to reduce apical dominance and cause lateral growth. Why not all flowers? Yeah, very good question. Baptist school, you are asking me why all flowers are not facing towards the sun as sunflower. A very good question, I'll answer it. The thing is, sunflower is a large flower. It produces more oxen. Most of the flowers are small, they produce less oxen. Sunflower produces more oxen. Lotus produces more oxen. So whenever more oxen is there, effect will be more pronounced. It will be clearly visible. If oxen quantity is less, then the effect will be very less. It is difficult to observe it. But all flower buds change into flower, opening of flower bud into flower, it is due to oxen only. Okay. But for opening and closing and facing the sun, we require large amount of oxen and large flower. It is only possible in case of sunflower, lotus and some other plants as well. Okay. That is the reason. A very good question. I hope your doubt is cleared. Okay. Now there is one more interesting thing. Oxen has an effect. It reduces the leaf fall, fruit fall. It reduces leaf fall and fruit fall. So how can we use it for our daily benefit? We can use it for a benefit. We can use it to a benefit in this way. Now you have seen apple orchards. Apple orchards, any fruit orchard, there is always a problem. Fruits keep dropping down at premature stage. They are not ripe yet they are dropping down. So to reduce this problem, gardeners spray a mixture of oxen and cytokinin called pomalin. It reduces the fruit fall in any orchard. Okay. Oxen delays senescence and aging. Senescence and aging is the same thing. There should be E. I'm just writing it short form. AGEING. senescence and aging. It reduces fruit fall, leaf fall. It reduces fruit fall, leaf fall. So gardeners use a mixture of auxin and cytokinin to reduce premature fruit fall. Okay, so you can use it if you have an orchard nearby, if you have a fruit orchard nearby, if you have fruit trees in your home, you can spray a mixture of auxin and cytokinin. It is available in the market, okay, and you can try it. You will be very happy with the results to reduce premature fruit fall. Okay, so that is what we do, all right. And there is another thing, auxin and cytokinin, they are antagonistic to each other. Auxin and cytokinin, they are antagonistic to each other. Cytokinin are opposite to each other or antagonistic. Okay, I'll explain. Auxin has apical dominance effect, cytokinin has lateral growth effect. Okay. Actually, I suppose this chapter would be interesting to you because the applications of plant hormones are so much. Okay. I wish to tell you more about this, but I wonder if you will be bored. There are some synthetic auxins which are used as weed killers. Okay. Maybe you would be interested, maybe you not be. This question was asked. Okay, so cytokinin. Cytokinin, the chemical name is 6-for-furyl-aminopurin. Let's not go into the chemical name. It is naturally found in. It was discovered from coconut milk. Coconut milk. Cytokinin. Cyto means cell, kinin means to increase. Cytokinin is a plant hormone, second plant hormone, which increases the rate of cell division. This plant hormone has the property of increasing rate of cell division. So what will happen if cells are dividing? Plant will become more healthy. Okay. If you have a fruit tree, suppose you have a guava tree or an apple tree or maybe a mango tree. I'm not sure mangoes are freely available in Northeast. Not all regions have, not all Northeast regions have many mango trees that I know of. You have many fruit trees otherwise. Anyway, if you spray cytokinin on a tree, what will happen? Lateral growth will increase, lots of branches will come, more branches, more fruits, more benefit. Okay, and any plant which is in a dying stage, it is not reproducing, it is not having a proper cell division rate. It's in a very in a very good condition. Its leaves are yellowing. If you spray cytokinin, it will suddenly become green. Okay, this is the property of cytokinin. So how do you get cytokinin? Take coconut milk and spray on any plant. You can see the effect in a few days. Okay. So it was discovered from two sources. Cytokinin was a plant hormone. It was discovered accidentally in two sources. One was maize seed. That is why another name of cytokinin is zeatin, and other source was coconut milk. And also sperm whale the sperm whale fat was actually the first source of cytokinin. It was undergoing pyrolysis. It's a term in chemistry, and cytokinin was accidentally formed. Okay. Anyway, let's not bother you with so many details. Cytokinin has the property of increasing cell division. It increases, it causes growth of lateral buds. Where can you get coconut milk? Coconut milk is nothing other than coconut water. Coconut water is equal to coconut milk. Okay, they are one and the same thing. Anyway, all right. But please don't spray coconut milk directly. Mix it with some amount of water, otherwise ants and a lot of other insects will come on the plant and start feeding on the coconut milk. You will get negative results. So a small concentration is required. Do not spray a whole coconut on a plant, okay? Don't do that. All right. So it causes growth of lateral buds, all right? Now, the biggest application of cytokinin is in tissue culture. Biggest application of cytokinin is in tissue culture. In tissue culture. Do you know what tissue culture is? Do you know what tissue culture is? Yes or no? Do you know what tissue culture is? Tissue culture is based on a principle called totipotency. Totipotency is the ability of any plant cell to grow into a full plant. What we do, we take a plant, take some cells from the plant, put it in a mixture of cytokinin and auxin. It grows, multiplies, cell division is induced, cell elongation by auxin, cell division by cytokinin. So it forms a mass of cells, circular mass of cell. We cut this mass of cells into many pieces, okay? And then we take one piece, we dip it in cytokinin and auxin mixture and increase auxin. Root is formed, then shoot is formed, full plant is formed. Now we can make hundreds of plants in a short time by help of tissue culture. It is not clear. Okay, give me some time now. All right, let me tell you where tissue culture will be used. You know banana trees, okay? Banana trees you know well. Do you know what is the ripening time, like flowering, fruiting time of banana? How much time does it take for a banana tree to grow from a seedling, a small plant, a plantlet, to a full plant and bear the fruits and flowers? Do you know? 6 months. But by tissue culture, we can make banana plants which give flowering, fruiting in only 3 months. Okay? There are some plants in the northeast which were endangered. There are some very beautiful orchids which were considered endangered. Why? Because orchid produces the smallest seeds known in nature, and they were not reproducing very properly. So what did scientists do? They used tissue culture to multiply the beautiful orchids found in Sikkim, found in the outer regions of the Nagaland region. There are very beautiful orchids, especially Sikkim. Okay, they multiplied the orchids present in the forests. Okay, these orchids have a special property. They do not grow their roots in the soil. Instead, they grow on the trees. These are called epiphyte orchids. Do you know about them? You people are from the northeast. Do you know about these orchids? Very beautiful orchids. They are very peculiar shape of them as well. One is called the monkey orchid. It has the shape of a monkey. Another is a lady's slipper orchid. You know, sandals and all that. There is an orchid which look exactly like the footwear of girls. Okay, it's called the lady's slipper orchid. Very beautiful orchids as well. These are the peculiar ones. They are very beautiful orchids. The maximum percentage of orchids is found in northeast region in the whole of India and Asia only. Yeah, very good. Yeah, very good that you know about it. So these orchids have very small seeds and they have no endosperm. So it is very difficult for these seeds to grow naturally, very difficult. So that is why we have used tissue culture to increase the population of such plants. Okay, any plant which is an endangered category, we can use tissue culture to produce large number of plants in a small time. Okay, another application, there are some plants which are very much desired by people. There are some plants which are very much desired by people. One such plant is seedless watermelon. Seedless watermelon. Seedless banana you already know. There are some plants, some banana trees which have seed, but there is seedless banana. The banana which we eat is seedless. Now people want to eat seedless watermelon. Can you imagine how good it will be to eat a watermelon which has no seeds? It will be wonderful, right? Now what is the difference between watermelon and banana's mode of propagation, mode of reproduction? Would you like to eat a seedless watermelon? Yes or no? I believe I have told you about it. Okay. But there is a problem. Watermelon which has seeds, it is very easy to grow. You eat the watermelon and throw the seeds in the field. But seedless watermelon has no vegetative propagation means. Now this is a problem. Banana has a vegetative propagation method. You grow a banana tree, there will be small trees which grow at the base of the plant. These are called plantlets. They are called sucker plants. Small plants will appear on the base of the banana plant. Just a second. Now what is happening? In watermelon there is no such way of reproduction. It does not have vegetative reproduction. So if we make seedless watermelon, we can make it. There is a problem. We cannot make it again. There are no seeds. How will you grow it? There is only one way to grow it. Use tissue culture. Okay? Please share with us. Okay, I have seen it. There are two varieties of seedless watermelon. One is yellow in color, other is red. I'm just going to draw the diagram of tissue culture right now. Okay, I will explain to you. That is why I just wanted you to know what tissue culture is before I begin it. Okay. Now, suppose this is a plant. Okay, there is a plant growing here. Okay. Now it has some roots and all that. It has some roots and all that. Now, what we are going to do, we are going to take a small sample. You can choose anywhere, any part of the plant, but we do not prefer the roots because roots are contaminated with bacteria. Which part is best? It is the apical meristem part, this part, which contains apical bud. There are small leaves here, small growing here. We take small cells from this part. We can also take from the leaf part. Any part, you take a small tissue sample. You take a piece of forceps, you pluck a small leaf, okay? Take out a small leaf, a very small portion, not greater than this part which I am drawing on paper. It's hardly this much big. It's very small, right? We only need this much tissue sample from any part of the plant. It can be leaf, it can be flower, it can be any part of the plant. We can take even spore mother cells, or we can take androecium, we can take gynoecium, anything you can take. Now this part is taken out and it is put in a agar-agar solution. Do you know what agar-agar solution is? Agar-agar solution. Agar-agar solution, do you know, students? What is agar-agar solution? It comes in green alga, sorry, red alga, sorry. Look, there are two red algae, okay, Gonialax and Gymnodinium, and there are some other sources as well. But agar-agar is basically a product of red algae, and we take this out. We mix. Agar-agar has the property that when it is in hot condition, it will be in liquid state. But when you cool it, it will solidify, and it is transparent. So you can imagine agar-agar as transparent jelly-like substance. Okay, transparent jelly-like substances. In this jelly, we mix the nutrients, and we can grow bacteria, we can grow plants, we can grow animal cells. Okay, except virus, we can grow anything. Virus we cannot grow in this medium, in nutrient medium, but everything else we can grow. It can be bacteria, it can be any cell, any plant, any animal. We can grow it. Okay. Now, plants are easier to grow. Animal cells are very hard to grow. The reason is animals are more complicated and plants are less complicated. Now, this ability is called totipotency. You take small tissue size sample, okay, and I will make a clone of you. Okay. So what I will do is I take a small pin, I put it on your face, or I take a cotton bud, and I use this cotton bud to take a tissue sample. Okay. First I pluck your finger with a needle. When the blood will come out, I will take this blood. Okay. Then I will put it in a nutrient medium, and I will make a clone of you. Okay. So I am not able to recognize you, people right now by the help of camera. I cannot see your faces very well right now. Otherwise, I would have called you by name. Okay, that I cannot do unless I visit you people. Anyway, so suppose the girl who is sitting at the back. Okay, I call you, I take your sample, and after about 15 days or 1 month, using sufficient nutrient and tissue samples, I will make a clone of you. Okay, so this is called culture. This is called tissue culture or animal culture. It can be plant or animal. Okay, right now it is not possible to make human clones, but we can make plant clones. Okay, so please do the camera also. Okay, so right now it is not possible to make animal clones, but we can make the clones of plants. Some animals can produce clones, but humans, it is totally impossible to make clone right now. Okay. Otherwise, if there were clones, you can ask your clone to come to the class, attend the classes, give the examination, top the examination, then give the seat to you. Okay. It would be so good, right? Just imagine about it, how good it will be, right? One can go for 1 day, other can go for 1 day. Okay. You can miss the classes. There will be so much time on your hands, and you can do so many things if your clones were there. Okay. Anybody who watches Naruto remembers what shadow clones can do, right? The Nan-Batte Dayo technique. Anyway, so let's continue. So what we are going to do here, we are going to take a leaf cell. We can take a small leaf, take some cells out of it, and put it in a nutrient medium. And this nutrient medium, agar-agar nutrient medium composition is agar-agar nutrient medium containing auxin and cytokine. Auxin and cytokine. All right. Auxin and cytokine. Now we take it out. Okay. And we put it in another medium. Okay. If sorry, one step has been missed. If you allow this tissue sample to grow, then after some time you can see that this tissue sample, which was small in size, has grown large in size. It has suddenly increased in the size. This increased tissue sample size is called callus. Callus. Okay. Cells are undergoing cell division, so it was very small. Now it has become big. Okay. Now this is called callus. Now we cut callus into many parts. Suppose you want to say 16 parts. Let's cut it into 16 parts. So you can cut it into a pizza shape pattern, 16 parts, 8 parts, 4 parts. Now each part can produce a new plant. Each part can produce a new plant. Each part can produce a new plant. Okay, I take this one tissue sample and I can use it to make a new plant. Okay, I will use every part actually. All these parts 1, 2, 3, 4, 5, 6, 8, 7, 8 or 16, whatever they might be, 1, 2, 3, 4. Okay, I will use all these parts. Now I will put it in a separate medium. Separate medium. Now this has a different composition. Previously auxin in this agar medium, auxin by cytokinin ratio was 1. Now I will increase the auxin ratio compared to cytokinin. So auxin upon cytokinin ratio will become more than 1. In this condition, roots will be formed. Okay. In this condition, roots will be formed in this callus. Root will be formed. But shoot is not formed yet. Shoot is not forming. Now I will take more cytokinin, less auxin. This will cause shoot initiation. First of all, root is initiated. Now shoot is initiated. Now the upper part is formed. There is a small plant growing in a test tube. You can call it test tube baby. Okay. So there is a small test tube plant baby here. Okay. And now you put it in a field. We actually transfer it to greenhouse. Transfer to greenhouse. Transfer to greenhouse and let it grow in the soil, and now it will become into a full plant. Are you getting me? Transfer it into a greenhouse. Now it will transform into a full plant. So this is how it is done. Okay. So we have taken a small tissue sample and it has grown into a full clone. So this is a cloning technique. Tissue culture is a cloning technique. We are making clones. 16 clones from one tissue sample. Okay, 16 clones from one tissue sample. So this is tissue culture. Which hormone is responsible? Most important, cytokinin. Without cytokinin, we cannot do tissue culture. I can give this to you separately. This diagram is not proper. I have drawn it in a very rough manner. But everything is correct, how it is done. This is the correct procedure. Okay. The question is, in 3 years ago, there was a question asked, why do we take apical meristem in tissue culture? The reason is apical meristem is virus-free, pathogen-free. If you want to write, you can write. It's up to you. Okay, but I'll move according to the time that is available. Okay. All right. So I hope you have written it. All right. I'll write it if you want to know. Why do we take apical meristem normally in tissue culture. In tissue culture. Because we do not want other bacteria pathogens to grow. Okay. Because apical meristem is always pathogen free. Why do you want pathogen free? Because if you take any other part and it has pathogen, along with the plant, bacteria will also start growing in the tissue culture medium. They will contaminate it. We do not want the resources to go to the virus and the pathogens, the bacteria. We want only the plant to grow. So we take the apical meristem because it is always pathogen free. Now this will be a separate question, why it is pathogen free. This is your homework if you want to know. I have been accused of making the class boring. I do not want to bore you so much. Okay, this will be a separate question. If you want to know, you can Google it. You can try to know why it is pathogen free. There is a reason for that. Okay, there is a reason why it is pathogen free, because it's in a constant state of cell division. If it was infected, it would have been destroyed. If it is in a constant state of cell division, it is growing, then it means the apical meristem is fine and okay. It does not have any virus and pathogen. If there is any disease, it will be passed on to the lower cells, because apical meristem cells do not have reserve food, so there is nothing in which bacteria pathogen can grow. Okay, other cells are fat. They are large in size. Bacteria pathogens want resources. They will grow in those cells. But apical meristem, next week you will be class 12th. Okay, apical meristem is always pathogen free. So please remember it. It has come 3 years ago in a question. All right, let's move on further. Okay. Now after that comes a hormone called gibberellin, third plant hormone. First is auxin, second is cytokinin, third is gibberellin. Gibberellin. Now from gibberellin, a question always comes. Gibberella was discovered from? The answer is Gibberella fujikuri. There were 2 Japanese scientists. They noticed that there is a strange disease in the rice seedlings. What is this disease? In this disease, the seedlings grow abnormally tall. They grow tall, and what is the problem with tall seedlings? They will, once there is a wind blowing, they will immediately fall to the ground. There will be no growth. You can understand what is the problem with tall plants. They have to face the wind. If they do not have mechanical tissues, they will fall to the ground. Does rice seedling and rice plant have mechanical tissues? Yes or no? People from the northeast, we have seen a lot of paddy fields, rice fields. You know this very well. My question is, does the rice plant have mechanical tissues, strong mechanical tissues? Mechanical tissue means wood. Okay, come on. It does not have. Yeah, it does not have strong mechanical tissues. It has small amount of sclerenchyma. It is not sufficient for the plant to grow tall as a tree. This is why some plants are small. This is why some plants are tall. Tall plants will always have strong mechanical tissues. So what will happen to the rice? It will fall down. So they noticed that this disease is, they gave this disease a name called foolish seedling disease. Gibberellin was discovered and isolated. Let's write now. Was discovered and isolated from Gibberella Fugicori. Okay, which causes Bakini disease. Bakini is a Japanese term. Bakini or Bakini. Bakini disease. Foolish seedling disease. Foolish seedling disease. Now, because it was discovered from Gibberella, so it was called gibberellin. And what is the effect of gibberellin? Now I must tell you. You have seen grapes. Now there are some grapes which are of the long variety. Have you seen those grapes which have long? There are some grapes which are round. Now there are some grapes which are particularly long. I'm not sure if you know about it. Look, I live in Delhi. I know about these hybrid grapes. Actually, they are not hybrid. I will explain. Chapter diseases from next week. Are you referring to immune system? Okay, eaten a lot. Now I will tell you. Gibberellin has the property of increasing the starch conversion to sugar, okay, and metabolizing everything and causing cell elongation. So if you spray gibberellin on a bunch of grapes, each grape will grow larger in size, it will be more tasty, it will become sweeter, and it will increase in volume. Okay, these grapes are particularly the long variety. It is particularly sweet in taste because it has been sprayed with gibberellin. So gibberellin is the most used hormone in a fruit orchard, particularly in case of grapes. Okay, so gibberellin, I will make it short. There are some other details. What is the chemical composition of gibberellin? It's called gibberellic acid, Ga1, Ga2, Ga3. You will just write the effect. Gibberellin has some effects called, I will write the effects first. Breaks seed dormancy. Seed dormancy. Increases cell metabolism. Increases cell metabolism. Okay, and converts starch into sugars. Converts the starch into sugars and increases fruit size. Increases the fruit size. Okay. So if gibberellin is sprayed in a fruit orchard on a bunch of grapes, the grapes will automatically increase in size. Gibberellin. Okay. They will become larger, sweeter, better. They will increase in the size. Particularly, they will have an effect of becoming longer. Okay, these are more sweeter. So this is the effect of gibberellin. Some long varieties of grapes are also there, but the hybrid varieties are always produced by this method, raised in this method. There is another effect, and this is the most asked question about gibberellin. Okay. It is called bolting. If you see the previous year question papers, you will always find every year almost there is a question on bolting. Bolting is an effect in a test of gibberellin. What is bolting? We have seen cabbage. Okay, cabbage has a closed inflorescence. Okay, sorry, closed bunch of leaves, and the what happens here? The stem is very compact. Internodes are very short. Internodes are very short. Now what happens here in cabbage? If you spray gibberellin, suddenly the leaves will start separating from each other. Internodes will become longer. Plant will become long, and in the center there will be a flowering axis. In the center there will be a flowering axis which will bear the flowers, the flowering axis or the fruiting axis, whatever you want to call it. This effect is called bolting. It was short, now like a bolt of lightning, it suddenly became tall. Okay. Alright. So this effect is called bolting. So I've scrolled up Senapati students. I hope it is more clear and visible to you now. Okay. Alright. Okay. I hope it is visible, right? Government Visa School. You have a question from me. What is the meaning of Gayatri Mantra? It's the meaning of Gayatri Mantra translates into may we go towards light from the darkness. May we be guided. It's a prayer which says, let's move on from darkness to the light. Okay. Why are you asking this? I'm not sure. Anyway, bolting, this is very important. Bolting, you can see previous year question papers, there is always a question on bolting, and bolting is observed in cabbage. I can show you some images after class. In cabbage, after spraying gibberellins, the plant suddenly changes in appearance. Changes in appearance. Now, what are these changes? What are these changes? Let us see. I will draw the figure. Normally, cabbage is of this type. The internodes are very close to each other. They are very short. Alright? The internodes are very compact. They are covering each other. Where can we get gibberellins? You can synthesize it artificially, or you can get it from the market. They are available. Okay. You will have to go to a shop which sells chemicals related to agriculture. There you can get it. Now I'm not closing this figure. You can understand it. One leaf is covered by other. One leaf is covered by other. This is the manner of arrangement of the leaves. Internodes are very small. Now if you spray gibberellins, I will also draw the root. This is the root of cabbage. It's a bit elongated. Okay. So spray the gibberellins. Now what will happen? It will suddenly change in appearance. It will suddenly change in appearance. The leaves will become separate from each other at a great distance. Okay. On the top, there is a flowering axis. The inflorescence axis is at the top. This usually happens naturally when season of cabbage is over. So right now, if you go in the fields and cabbage is remaining, you can see some of these plants growing like that. If you have cabbage in your yeah, you can use it on strawberries. Government Mizo School, you have asked me a question. What can I spread to my strawberry plant so that my strawberries become larger? Look, normally for strawberries, gibberellin can be used, but why do you want to? Okay, if you have the wish, you can spray gibberellins. You can spray gibberellins. Okay, they will become larger. They will become larger and sweeter. Inflorescence axis, I'm not drawing here. Okay. Does the change happen immediately after spring? Look, please try to understand this. The hormones always have an effect. This effect is seen after many days. This effect is seen after many days. For example, in our body, the reason for an increase in the height is growth hormone. Okay. The reason for the increase in height is growth hormone. Now, if somebody has higher concentration of growth hormone, he will become taller, and whoever has lesser concentration, he will not be as tall as the other guy. But these changes have not happened in a day. For example, the appearance of secondary sexual characters. Okay. For example, when you crossed 12 year of age and came into 13th year, okay, that was the age of puberty. Some people might have it in the age of 11 or 12. Normally it is between 12 to 13. So what happened? You suddenly woke up and your mom failed to recognize you, that you suddenly have a beard or you have a mustache and your appearance have changed and your voice has changed. If this has happened, our parents will stop recognizing us. That who are you? What are you doing in my house? This is not my son. This is not my daughter. Okay. So these changes have not come overnight. For example, in the case of beard, slowly, very slowly, because of the effect of testosterone, the hair which were always present on your face, they have become more thicker. They have become more thicker. There is more melanin in there. So that is why it becomes the reason for the beard or the mustache. Slowly, they have become harder, stiffer. Okay, they will increase in the diameter. They were always there. They were always there on your face, but suddenly in puberty they have become changed in appearance. Or in the case of underarms also, this is the same story. Hormones produce an effect which is going to spread over years, days, or hours. Okay, so it is not going to happen overnight. The same is the case of gibberellin. You cannot observe it immediately. You must give it 7 days time, or in 7 days you can observe the effect of gibberellin or any hormone. Okay. So where should we spray the strawberry? In the strawberry or on the plant? Of course, you should always spray it towards the strawberry. If you spray it on the leaves, the effect will be a bit different. I do not want to go into the details, but normally it is sprayed on the fruits. Normally it is sprayed on the fruits and the fruit axis. Okay, all right. So this plant which was dormant, okay, cabbage was dormant. Now it has suddenly changed in appearance and it has undergone flowering stage, fruiting stage, and the leaves have separated from each other. This happens in the month of February when the winters are over. This happens in the cabbage plant. Maybe you have seen it already, maybe you do not remember it. Light yellow colored flowers come on cabbage or light white colored plants. Okay, so you must have seen it, maybe you do not remember it. There is one more effect. Germinating barley seeds. Question is coming. Germinating barley seeds contain which hormone? Germinating barley seeds contain which hormone? The answer is gibberellin. Okay, I'm teaching you gibberellin, so the answer will not be different. Okay, gibberellin. I told you why, because this is so because gibberellin breaks seed dormancy. So if a seed is in the dormant state, okay, it is in the dormant stage, the seed will have the concentration of abscisic acid, ABA. But if it is going to break the dormancy, it is germinating. Now this seed will contain gibberellin. Okay, that's what is going to happen. So any germinating seed will contain gibberellin. Okay. Now, what is the effect of gibberellin on barley seeds? It will produce more starch, more sugars. What is the use of more sugar in barley seeds? If it is going to produce more sugar, more alcohol will be obtained. So germinating barley seeds are soaked in water. Now seeds start producing gibberellin, and if we soak it overnight, soak it for a long period of time, sufficient long period of time, all the starch will be converted into sugar. Then we use yeast to convert sugar into alcohol. Okay, this is the use of barley seeds. Alcohol, normally beer is made from barley seeds. Okay, all right. So the last two hormones, we must finish it. Why alcohol? Yeast undergoes, I already told you yesterday, yeast has the property of changing sugar into pyruvic acid, and pyruvic acid is changed into alcohol, ethyl alcohol. Okay, so in the alcohol industry, this is the use of gibberellin. We do not normally spray gibberellin. I'm just telling you which hormone is present in germinating barley seeds. Gibberellin, which converts the starch into alcohol. You can also say germinating wheat seeds, which are used to make vodka. That is also the same principle. Or from a potato also, same principle. Same principle. Okay. Normally, whenever a seed is germinating or any plant part is germinating, there will be the role of gibberellin. Okay. Okay, all right. So let's move on to abscisic acid. Now remember, I told you auxin and cytokinin are having opposite effect. Similarly, gibberellin and abscisic acid have the opposite effect. This is the fourth hormone, abscisic acid. This hormone is present in dormant seeds, and dormant plant parts. And dormant plant parts. Okay, so yeah, you are asking me, government meso school, sir, can humans also produce alcohol? No, we cannot produce alcohol. The enzymes for making alcohol are absent in our body. Instead, our muscles can produce lactic acid. We can convert alcohol into other products. In our body, alcohol is converted into first acetaldehyde, which causes hangover effect. Hangover effect is due to acetaldehyde accumulation in the body. Next, acetaldehyde is converted into acetic acid. Acetic acid is eliminated from body as urine in the form of acetic acid in urine. But if you want any person to leave the alcohol habit, there are some drugs which will cause delayed oxidation of acetaldehyde into acetic acid. This is a de-addiction drug. So any person who takes this drug, he will have prolonged hangover. The hangover will not go for 2 days, 3 days, 5 days. Ultimately, he will say, I am not drinking anymore. I cannot take this. Okay. So if you have any friend, any senior you know of that he is unable to leave the drinking habit, you can mix this drug. I will tell you the name. Ask me it. Ask me the name separately. Take this drug, mix it in his food, water, anything, and he will have a hangover lasting 4 days, 5 days. Okay. This is actually a de-addiction drug. I'm just joking. Do not give it to anybody. It is used by those people who wish to leave their alcohol habit. Okay, but you can also give it unknowingly to a person. Never tried it, but I know what effect it can cause. So that's your question, I believe. We cannot produce alcohol. Now, abscisic acid, it causes the plants to become adapted to adverse conditions. Adapted to adverse conditions. Adverse conditions. What are these conditions? Example, drought. Drought means water scarcity condition. Drought, okay, etc. A lot of things can be physiological, dry soil. A lot of things can be possible. Normally, we talk about adverse conditions like winters, no sunlight, or there is no water in the soil, drought. Okay, so this is responsible for the dormancy and the resistance of plants to adverse conditions. Okay, now how can we use it? How can we use it to our benefit? Now you know you are living in the northeast. Now there are some regions which are very dry, arid. Rajasthan, for example, in India. So there are some regions in Rajasthan which receive very less rainfall throughout the year, very less rainfall. Now it is very difficult or impossible to grow crops in such areas. So what is the problem? If you get water through the plants through the water canal, we can use irrigation systems or water tube well. Now in these regions, agriculture might be possible, but there is huge demand of water by the plants. So what can we do? We can spray synthetic abscisic acid. Okay, the name of one such synthetic abscisic acid is phenyl mercuric acetate. If you spray it, these plants will be able to grow in water scarce conditions. This is the use of abscisic acid. Okay, abscisic acid causes closure of stomata. Closure of stomata. ABA, synthetic ABA, sorry. Synthetic ABA is used in growing plants in water scarce conditions. Water scarce conditions. Okay. Synthetic ABA is used in growing plants in water scarce conditions. All right. Example, write the name of the compound. You might be asked this question. Such compounds are also called anti-transparent. Anti-transparent, which prevents transpiration or evaporation by transpiration. Anti-transparent called a PMA, transparent, called PMA, phenyl mercuric acetate. Phenyl mercuric acetate. Okay, this is the use of ABA. Now this is all about ABA that you need to know. Now the next hormone is ethylene. Last hormone is ethylene. Now when we talk about ethylene, I believe you have all, let's talk about a story. I will tell you a story. Now please tell me if you remember the story or not. When you were in the lower classes, you must have read about a story in moral science regarding the company of friends. The title of story is a man is recognized by the company of friends he keeps. It's a thought in moral science. Now maybe you have heard about it, maybe you have not. I'm not sure. Let's listen to the story. Now there was a man who had a son, and his son was very spoiled. Let's say he was very spoiled. He used to drink and smoke at a very early age, and he was not very happy with his son's habits. He came to know that his son is also engaged in gambling habit. So all these habits cannot be considered to be very good habits, especially at a very young stage. So he was not very happy. He knew that his son was very good, but later on, because of friends, he changed, and because of his friends, because of peer pressure, friends asked to do some things and he cannot refuse, or maybe something like that. He was sympathetic with his son, so he took him to the market and asked him to purchase apples of the best quality. Okay, apples of the best quality. So when he purchased these apples, he took one apple which was rotting, one apple in rotten condition, and he told his son to keep one rotten apple in a cupboard along with very good, a cupboard full of fresh, which were very tight apples, which are not going to spoil for a long time by left to themselves. Okay. In this cupboard, he put lot of good apples and one bad apple, one rotten apple. Now, within one day, he asked his son to open it after one day. And when he opened the cupboard, all the apples were rotten. Okay. All the apples had over matured and suddenly become overripe and rotten. They were not to be eaten anymore. So he told his son that this is the effect of company on friends, and his son undergone radical change, and he suddenly changed his company and started keeping good friends and all that. So the moral of the story is we are recognized by the company of friends we keep. Now why I'm telling you this story, the reason why all the fruits became rotten was one rotten apple, ripening fruit, produces a hormone called ethylene. If you put ethylene with other non-ripened green fruits of any kind, they will suddenly become overripe. They will suddenly start ripening. They will suddenly become yellow or orange or red, depending upon their natural fruit color. For example, apple is green. Later on, it will become yellow, red. Okay, and when it is rotten, you know which color it assumes. Okay, it's always red with a dark brown color of the flesh of the pulp. Now, this condition can be observed in anything. Take you can when you go to your home, you must have heard your mother saying, if you have a refrigerator, like, of course, you have a refrigerator. I'm joking. If you take a refrigerator, just wanted to tell you this example, and you have a poly bag full of tomatoes. I know very well there is a very less use of poly bags in Northeast. I know it. I'm just asking you people to assume. You have a poly bag or a packet full of tomatoes, and you close the top. Do not allow air to escape from the tomatoes. Keep it in the fridge or anywhere you want. Now open it after 1 day. Open it after, let's say, 12 hours. Go in the night. Open it after you are woken up. Now when you open this bag, suddenly you will find all the tomatoes are rotting. Why? Ethylene. Okay. If you want bananas to go from one place to another, bananas are always transported in green stage, never in the yellow stage. Otherwise, the other people will get only insects and crawling among the jelly of the over-ripened bananas. They are always transported in green stage. Now, in the green stage, what will happen is these bananas that are there, what is going to happen? In this green stage, the bananas, they produce very less ethylene or no ethylene. Now, if you place yellow banana close to some green bananas, they will quickly change their color. They will start becoming ripe. So, if you change the conditions, keep them in a well-ventilated space, they will not become ripe. You can transport them for 1 month or 2 months. Okay. Now, when you wish to eat them, you can suddenly put some ripened bananas with it. Within 1 day, they will change their color. Okay. This is what is done with ethylene. Sometimes when bananas come, you have seen there is a white colored chemical or something which is on the top of the bananas. It's a chemical called ethefon. It slowly releases ethylene. That is why our parents say, if you are taking some fruits, always wash them. Some chemical might be remaining which causes ripening. So we do not wish to eat the chemical, we wish to eat the fruits. It is the only gaseous hormone. You know the formula of ethylene? CH2 double bond CH2. CH2 double bond CH2. Okay. Only gaseous hormone. And it is produced by ripening fruits. Okay. For example, a pineapple which is undergoing ripening, it will produce a large amount of ethylene. Place a yellow pineapple with a green pineapple, it will suddenly change color. Produced by ripening fruits. It is used for fruit ripening when required. Fruit ripening when required. It is used for fruit ripening when required. All right. Another thing about ethylene, it is used for fruit ripening when required, and it is a gaseous hormone. So there is a chemical called ethefon. A chemical, brand name of chemical is ethefon. Is used in artificial. This question is asked. That is why I am labeling it. Any question which has been asked in previous examination, I always draw a hash. I always draw a asterisk or a hash that this question was asked previously in the previous year question papers. Ethefon is used in artificial ripening of fruits. Artificial ripening of fruits. Not much detail is required other than this. Okay. If you want to write something more, you can say, how does it affect the fruit? Ethylene causes senescence and aging. Abscisic acid causes senescence and aging. Auxin, cytokinin, gibberellin, they cause maturation, elongation, and cell division. But abscisic acid and ethylene causes aging, death, decay of a plant cell. Okay, that is why plant is changing the color. Now it has suddenly changed the color because of aging of the cells, old nature of the cells. Okay, is it dangerous? No, it is not dangerous. Please don't eat it. Of course, yeah, ethylene is not dangerous, but ethefon, it's actually not dangerous, but the effect is not very good on the health. All right, so it causes senescence and aging. Senescence or aging, same thing. Aging. Okay. Now there is one last point. This question is also asked. It is actually a question of auxin. Okay. Question of has come in the previous year examination. Name one artificial weedicide. Name one synthetic auxin. Question is asked in many different languages, in very different manners. Name one synthetic auxin also used as a weedicide. This is important, that is why I am labeling it in the end of the chapter. Also used as weedicide. Another question is, name of auxin, synthetic auxin used in Vietnam War. Vietnam War. Or what is Agent Orange? The answer to all these questions is one word. What is Agent Orange? These questions just for one thing. So many questions from just one part, which is called 2,4D. This is the name of the chemical. Yeah. Now there is a different question which is derived from it. Okay. 2,4D caused cancer in the American soldiers who were attacking Vietnam. Why do we use it? Look, there are two things. How does it work? In a wheat plantation or a rice plantation, there are a lot of weeds. Now, auxin has the property of causing growth. If we use synthetic auxin, it will also cause growth. So, how are we using a chemical which causes growth as a weedicide? Let me explain to you. Rice and wheat plants have very thin blades, thin leaf blade. Auxin is sprayed on the plant, on the leaf. So, wheat plant will absorb very less synthetic auxin. But the weeds which are growing in that field, they usually have broad leaves. They are dicots. If you spray synthetic auxin on a dicot, it will absorb a large amount of synthetic auxin. It will be actually harmful for that plant. It will grow large leaves, maybe large stems. Stem will become soft. It will drop down. It will suddenly drop down because of rapid growth with no mechanical tissues. So this way we can control the weeds growing in a wheat plantation or a wheat field or a rice field. Similar is the case with other things. Now what happened in Vietnam? Vietnam, what happened was, there are a lot of enemy snipers. You know snipers, right? Everybody plays Call of Duty or Counter-Strike or so many games. You know what is sniper? A sniper is a marksman who is especially trained for killing from a long range, and he uses a special rifle for killing. Now, Vietnam was using a Dragunov modified rifle obtained from Russia, and they were killing a lot of American soldiers. They were using guerrilla techniques. They did not come out in the open, they were hiding and killing. So what did USA do? It used Agent Orange. Agent Orange is not the name of some special soldier. It is the name of 2,4-D. 2,4-D was used in orange colored canisters. So it was called Agent Orange. It sprayed it on the warfare field. Whenever it was sprayed on the warfare field, all the plants died down. There was no place for enemy snipers to hide, so they were killed or they ran away. So that is how they started winning the Vietnam War, but actually they did not win it. They obtained a standoff and they signed a peace treaty. Let's go away. You go your side, I will go my side. But there was a problem. Agent Orange caused cancer in American soldiers. Why? This is the next question. 2,4-D, the name of it is 2,4-diphenoxyacetic acid. What happened is 2,4-D contained a chemical present as contamination. Contaminant 2,4,5-T. Contaminant. The question is, what is the name of the contaminant which caused cancer in American soldiers? First of all, if 2,4,5-T is given, you will write 2,4,5-T. This is the answer. Causes cancer and was present in Agent Orange as impurity. And was present in Agent Orange as impurity. Alright, this is the answer. So this is a different story how it got contaminated. It's a manufacturing process defect. It was not actually intended to be present, but a manufacturing defect caused this chemical to be present, and that is why it caused cancer in the American soldiers. So this is the answer. What is Agent Orange? Okay. No, it's not an orange. I'll tell you. There were large drums, orange colored drums. I'll draw the figure. This is orange in color, right? You can see it. I hope it's not appearing as a different color on your screen. Now what is written on this big drum? It is 2,4,5-D, 4D, orange colored drums. Now from aeroplanes, this drum content is going to be sprayed on the plants. Okay, from top they are going to spray it. This is Agent Orange. Agent Orange is the nickname given to these VDs, this VD side, which is helping the people indirectly. It is killing the plants, helping the people to find out where the enemy sniper is hiding. Okay, so it's a helping soldier, which is not actually a soldier but a orange colored drum. That is the reason. Okay. By the color orange, I just suddenly remembered one more thing. What is the color of black box? What is the color of black box in aeroplanes? It's also orange in color, but it's actually called a black box. Okay, so that will be all for this. That is what you needed to know in this chapter. In auxin, question comes from 2,4-D or synthetic auxin, how it is used in helping the plants. It prevents the fruit fall. Cytokinin question will come. It is used in tissue culture. Gibberellin question will come. Bolting, explain bolting, and which hormone causes bolting, you will write gibberellin. Which hormone is used for increasing the fruit size, you will write gibberellin. Which hormone is used in preventing fruit fall, you will say mixture of auxin and cytokinin? These are very important questions. Okay. And which hormone is used in agriculture to prevent loss of stomata, loss of water through stomata? Abscisic acid, synthetic abscisic acid. Exact name is PMA, phenyl mercuric acetate. And which hormone is used in fruit ripening? Ethylene. Okay, so that will be all. In the next class, we'll start with the class 12th syllabus, I believe. Now I have to give you notes and all those things. Okay, so thank you so much. Okay, and arigato. Okay, you know what I have written. Anyway, thank you so much, Shillong students, everybody. Okay, so good night. Okay, have a safe journey, reach home safely, have a good meal, and thank you for your time. Thank you.